Welcome everyone to this week's Wednesday at NICO seminar. I want to introduce our speaker, Aparna Baskaran. Hopefully I'm not doing a terrible job with the pronunciation there. She's visiting us from Brandeis University, Department of Physics, and also the head of their MRSEC, the Materials Research Science and Engineering Center. She got an early career award from the American Physical Society for soft matter research. She's also, I saw, won teaching prizes at Brandeis, and she's been a Scialog Fellow, which we have to talk about at lunch because I've studied the Scialogs actually. These interesting series of conferences by invitation only. And what I'll say, she's published in some of the top journals: Science, Nature, Physics, PNAS, Physical Review Letters, obviously doing great work. And I'm excited to hear about the active matter research that she's going to tell us about today. So welcome, Aparna.
Thank you. Thank you, Danny, for letting me come. And thank you all for coming to listen to me talk about some recent stuff that's happening in my life. Please interrupt at any point, and I'm happy to answer questions about life, universe, and everything, or technical questions associated with what's on the slide, or whatever works for you. Let me get started. The outline and scope of the talk, I just comically partitioned into three parts: a proclamation, some proselytization, and some prognostication. My proclamation is just the big picture framing of the research track. The proselytization is everybody stop doing whatever it is you're doing and solve my problems. I want to tell you what are the problems I'm trying to understand and how much I'm failing at them. And finally, the prognostication is some future things that I don't quite know how to do yet, but I just want to show you some of it. Okay, let's start with the big picture framing. I'm going through a phase in life right now where it's very hard to get out of bed. So I have to motivate myself. So when I do big picture framing, I mean big picture framing. I thought the big picture framing is what is the meaning of life? And so I asked Google, is that what I'm doing? And these are the answers it gave me for what is the meaning of life. To do good, to do the right thing, to leave the world a better place, and all of these self-help sounding words. And so I went into the scholarly literature and I found things like, meaning is a problem. So existentialism says, don't ask about meaning. Utilitarianism says, okay, we'll assign a meaning arbitrarily and things like that. So there's all of this philosophical thing. So clearly this is not what I'm doing. So I changed my question and I thought, okay, what I'm doing, I will call, what is the definition of life? Again, I asked Google, is this what I'm doing? And Google gave me answers such as this. And these are all from scholarly literatures in different fields. The period from birth to death of a living organism. And this sounds like life has the units of time, and unless you know the origin and the end, you can't know its value, and so on. It's crazy. I don't know what to do with that definition. The second definition it gave me was quality that distinguishes matter that has biological processes. And I was like, what the heck does that mean? What, pray tell, is a biological process? This seems like even worse than the first one. And then I came across this definition, which StatMech people like a lot. I'm a StatMech person. A self-sustaining chemical system capable of evolution. Yeah, okay, so this gives me some handle. I can take some network theoretical approaches to understanding this kind of a definition, but that's not my game. So what I ended up with was this laundry list definition of life: homeostasis, organization, metabolism, growth, adaptation, response to stimuli, and reproduction. This laundry list definition is what people use to decide if something is alive or not. So this laundry list definition gave me a sense of what it is that I'm trying to do. The game that I'm trying to play is to identify the essential principles for engineering living materials. And I want to articulate what I mean by essential principles and what I mean by engineering living materials over the next couple of slides. So what do I want to do? What I want to do is build myself a bag of crap, which when I press a button, can set up system-sized circulating flows, which is called cytoplasmic streaming in the biology literature, can go where I want it to go. This is motility, where you can see the middle movie is showing you neutrophil crawling and swallowing some E. coli. And I switch another button and it's able to set up mass transport and mass organization within itself in various ways. If this movie cycles again, this movie is chromosomal segregation, where the material is basically being sorted and arranged in space and time by what's happening inside the bag. I want to build myself a bag of crap that will do all of these things. That's what I mean by engineering a living material. If you look at it, it looks living, but I made it from bottom up. Okay, now for the purposes of what it is that I want to do today, I have a cartoon argument for why I'm working with biological systems and I want to build it bottom up from biological materials. The reason I want to learn from biology in a cartoon sense of the word is when biology decides to make the best football player, it makes this. Can you guys see Messi? Should I make it bigger? Look at him error correcting dynamically and accomplishing what he wants to accomplish and goal, right? Now, if I engineer this by just looking at what the organism is doing from the top down, what I end up with is this guy. This is the state of the art for football playing robots. This is from 3 years ago, so it's improved a little bit, but this is what football playing robots look like. When I watch a human play rope and then try to engineer it from top down, this is the level of engineering I can accomplish. So I absolutely and utterly want to use biology to build what I want to build from the bottom up because I think we can do much better that way. Okay, that's enough fun. Let's hide floating media controls and keep going. I apologize. I'm not anticipating the delay that it takes to show up there. Okay, for the purposes of today's talk, I want to focus on this one movie, which is associated with motility of a cell. And I want to show you a more modern version. So this movie is from Dave Roberts' lab in Vanderbilt University from 1957 or something like that. So it's a really old movie. But there are more modern movies of the same thing. This is again a neutrophil cell being watched under a microscope in the Bethsic lab at Genelia Farms. And what you will be able to see here is in green is visualized myosin motors, and in purple or magenta, what you're visualizing is actin. And what you'll be able to see is there's a lot of motor activity at the back end of the cell, not so much at the front end of the cell, and there's lots of polymerization activity in the front end of the cell and not so much at the back end of the cell. I'm a physicist, so I really think in terms of forces and momenta and Newton's laws. So if I think about it in that sense, what I'm getting is local contraction at the back end of the cell, I'm getting local extension in the front end of the cell, and that is producing global displacement of the cell. So I want to make myself one of these. So for the purposes of today's conversation, I'm just going to think about forces, and I want to think about how can I program forces to do what I want them to do. And what I learned by watching this biology movie is by learning that what I have some elementary units. In the particular movie, I'm showing the elementary units are myosin and actin. And what is happening is they are dynamically and adaptively reorganized. The motor concentration, the polymeric gel properties, the chemical reaction cascades are all organized in space and time in such a way that I end up with what function I want. So when I say I want to make myself one of these, I mean I want a material which has spatio-temporally programmable internal stress and hence will result in navigable motion. And I add a caveat to this. I want to put molecules in a cup and whatever this thing is should assemble. It should be self-assembled. So those are my macroscopic objectives. Now I'm pretty sure I will never get there before I die, but I want to keep walking the path as far as I can while I'm alive. So a theorist hatches a plan for doing this. I'm a theorist. I wouldn't recognize a protein if it hit me on the head. So I have to work with other people. And I have these collaborators, Guillaume Duclos, Seth Freyden, and Zvonimir Dojic, who have a simple active unit motif. The simple active unit motif has microtubules, which for our purposes, it's some polymeric protein structure, but for our purposes, it's a rod, which has a head and a tail. And it has a bundling agent. It is a microtubule associated protein that's called PRC1. We don't care. All it does is produce an attractive interaction between the rods so that they tend to line up when they are close to each other. And finally, there are motor proteins. These are guys that you have seen cartoons of where they step along one direction of the polymer, right? So those are the three ingredients. You put these three ingredients in a cup and you get active fluids. And we have many varieties of these active fluids. I'll show you a few as the talk progresses. But the basic active unit, this is just a time series of the basic active unit zoomed way in. Two guys come together, the motor proteins walk and slide, 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 slide and fall apart. That's the basic active unit. They're just rods come together, they slide, slide, slide and fall apart. So let's take this basic active unit, and then let's say I want to put a bunch of these in some circumstance, and I'll figure out a pattern of activity I need so that the material exerts whatever force I want on the rest of the universe. And I can't see how this can be wrong. We are not questioning Newton's laws. So if mass is moving, then there must be a force exerted by the object on the rest of the universe. Of course, it can't be a force monopole because I can't tear myself into two pieces, but it can be a force dipole. Right? So there's some force distribution, and that I should be able to engineer is my claim. So how hard can it be? I thought how hard can it be about 7-8 years ago. Now I know it's very hard, right? But I've decided to do it, so I'm going to do it. The circumstances under which I want to do this, for the moment, I'm going to forget about my bag, I'm going to forget about my cell, and I'm going to start in a very, very well-studied system in this field, which is the 2D active pneumatic. What you do is you take a bunch of these microtubules and you centrifuge them onto an oil-water interface. So they form a 2D film. And what that 2D film does is this sloshing, ice cream churning dynamics that is shown in the movie on the right-hand side. Okay? All right. This is very well studied. There's more than 10 years of theory literature on this topic, and there is some understanding that has emerged over the time that we've been working on it. Now, let me articulate the challenge, and I want to articulate the challenge in the following way. What I usually get by way of data, I want to build this, right? So I want to start with data, and what is the data that I get? It will be some movie a collaborator hands me. They make a movie under their microscope, and they hand me a movie. And what the movie gives me is an intensity as a function of x, y, and t. So I get a spatio-temporal map of intensity from that movie. That's my starting point as data. And the dimensionality of the data is not very large in the scheme of other data sets, but it's very large for a physicist. This is way too many data points. It's 10 to the 4 times 10 to the 6. So the 10 to the 4 is the pixel resolution associated with the field of view, and 10 to the 6 is the typical time series length of what one can get in these experiments. And what I want to do is control this system, right? I want to program this system, by which I mean I want to control the forces in my system. And the 3-step process to control, which anybody you stop in an engineering department will tell you if you're going to do data-driven control. The first step is compression. Change the high number of variables into as few as possible without loss of fidelity. Then build a forecaster, predict what will happen in the next step in real time, and implement a control. Change the driving so that the system will go where I want it to go instead of where it would have gone without the control, right? Those would be the 3-step process. Let me first do the 3-step process as a physicist, then I will come back to the data and see how I would do it in the data. So let's start with the first step, compression, but a la human, not a machine learning network. Compression for a human means as few symbols as possible in order to get to where I want to go. So the dominant textural signature that you can pick out when you're watching one of these movies is called a +half topological defect. So those of us who grew up in physics disciplines, the moment we see a topological defect, we know, oh, symmetry. Now I know how to work with this problem. So you start with symmetry and conservation conditions. And for that, what you basically need to do is start with a force dipole that lives in some dissipative medium and add to this aligning interactions. The force dipoles, when they come close together, align with each other. Okay? If you take these two ingredients, then what turns out to be the 2-symbol description, it's not 2 symbols, it's secretly 9 symbols, but it's 2-symbol description of the system, is a nematic order parameter, which is a traceless symmetric tensor, which characterizes the alignment of these double-headed arrows in any region in space. And then there's the velocity. These things are moving, so I have a fluid velocity associated with it, so I have a velocity field. So I have these two fields in space and time as the symbols. This is what compression means to a physicist. And once you figure out what symbols you want to write, it is pretty straightforward to write down the equations. These equations are very, very well studied in the literature. They are called active pneumatic hydrodynamics. I really don't want to spend my time walking through the derivation of these equations from physics principles. So I'll just tell you a one-line story about three-line story about these equations. The first important piece of this equation is the term in my Stokes equation, which I call the active stress. There is a parameter alpha that basically tells me how much this motor activity is going to drive flows. Okay? I have some motor activity that is captured by this parameter alpha, and that is going to drive flows. So I just have a source term in my Stokes equation that is driving flows. Then these flows move around those force dipoles. So the force dipoles get convected by the flow, get rotated by the flow, get shear aligned with the flow, and things like that. And in addition, to regularize the theory, at short distances, you have to add this molecular potential, which is just a free energy. It's just a gradient descent in some parabolic landscape. Okay? Those are basically the ingredients that I need for to capture the theory. For those of you who are curious, the form of the free energy is basically a phi to the fourth theory of some sort, but you have it's a first order transition, so you have to keep Q cube and you have to keep the Frank elasticity. And all of those words don't have to mean anything to you. So ignore all of that if you don't know what I'm talking about. Okay. The basic phenomenology of these equations, explained by way of cartoons so that we don't have to spend time with the equations, is as follows. Imagine that I have a completely aligned region in space. That is to say, all my force dipoles are standing one on top of each other in straight lines. So whenever I'm drawing one of these large black double-headed arrows, I'm not denoting one little unit. I'm denoting many, many units all lined up together. It's a coarse-grained arrow. So when these units are all lined up like this, every force exerted by one guy is cancelled by the other guy. So the net force inside the domain is 0. And the only force that I can find is at the edge of the domain. The gray regions, I call the rest of the universe. I don't care what is there. Whatever is there is being pushed upon by whatever is inside the domain I'm watching. Okay? So this is force balance. This system should nominally stay there forever because I have perfect force balance and everybody is at rest. But if there is a small fluctuation in the alignment in any region, again, I'm not bending any single rod. I am bending the line of rods. The line of rods bends a little bit, like people standing in a coffee line. The line can meander without you squishing any one person. The line meanders. Then what I see is that the net force exerted on the rest of the universe is no longer normal to the rest of the universe, normal to the interface, but actually there's a tangential component. The tangential component of the force, if you do a little trigonometry, you'll convince yourself it can be represented by this red arrow. Okay? And I don't know what is true or not true in this world, but Newton's third law cannot be wrong. We will be terribly screwed if Newton's third law was wrong. So Newton's third law tells me that these guys, the rest of the universe, will exert an equal and opposite force on me that will be denoted by the green arrow. So what do I know? When one of these domains bends a little bit, it will also start moving. Right? Because it will respond to the green arrow acting on it, so it will start moving. So the domain bends a little bit and it starts moving. But also I'm pressing on the domain from both sides. So if you have a floppy object and you press on it on both sides, it's going to buckle. It's going to buckle, right? So it's going to buckle and buckle and buckle until such time as I need to buckle a single rod. When I have to buckle a single rod, I cannot. The rods are pretty rigid on the scales that we are thinking about. What you get are these topological defects. Topological defects are structures that form in this order parameter field whenever I have to buckle one rod. Instead of buckling one rod, they just arrange themselves in such a way that they avoid the buckling. And these things are called plus half and minus half topological defects. Okay. All right, this is the story of the equations that I showed you, linear stability analysis of those equations, but in the form of cartoons. Okay. Now, I'm hoping that by watching this movie, you can see some of it. So there's this thing buckles and a plus half forms. So this thing now buckles and a plus half forms. So that's basically the dynamics that you're watching in the movie. And by analyzing these kinds of data, there are 3 groups in the world that produce a lot of data on this topic, and all of us theorists wait with bated breath for the next batch of data, and we've analyzed it quite a bit over the years. Excuse me. Sorry about that. The conventional wisdom says that I have some active stress, which is some parameter alpha that I don't know yet, and is proportional to the nematic director in any region. It is proportional to how ordered the rods are in any region. And this active stress is balanced by reaction stresses that come from the rest of the universe. The rest of the universe is again more of the fluid. So the reaction stresses are coming from viscous stress that the fluid can develop and the elastic stress that can arise in a nematic ordered system. And by balancing the active stress and reaction stress, you can get a length scale. That length scale is square root of k over alpha. And if you measure any length scale in the statistical steady state of the system, the length scales that you can measure are defect density, vortex sizes, and things like that. They all scale with this length scale. And if you measure any time scale in the problem, you can measure speed, you can measure velocity autocorrelation time, you can measure order parameter autocorrelation times. Any time scale that you can measure in the experiment always scales as 1 over alpha, where alpha is the activity. So it seems like there is an intrinsic length scale and an intrinsic time scale that the dynamics has, and everything scales with this quantity. And I would imagine that the driving, which is given by this parameter alpha, should be a function of the concentration of whatever crap I need to make this thing work, right? And there's a lot of crap. There are, I pick 3 here, which are the ones that we keep track of in the theory. KSA is the name for the motor protein. So KSA within square brackets says motor protein concentration. PRC1 is the bundler, so that's the bundler concentration, and ATP is the fuel. It's the fuel that makes the motor work. So those are the 3 things that we can kind of keep track of, that the experimentalist can kind of control. There are 6 other chemicals in the cup that form an ATP regeneration cycle and keep the motor from degenerating. I ignore all of that. They're all small molecules. I assume they don't do anything, and I ignore all of that for now. Okay? It will probably come back and bite my ass in a few years, but right now I'm ignoring all of that. Okay. So now I have a system. I described the theory of the system in terms of PDEs, and now what I want to do is tell you how am I going to control this system from a physicist's perspective. And this is work done with a recently graduated student, Saptarshi Ghosh, who just started at Broad Institute like a couple of weeks ago, and my buddy, Mike Hagan, with whom I do all of my work on these active systems. And what I want to do is, this movie is a numerical solution of the PDEs that I flashed at you a couple of slides ago. I just write some solver. It's this particular simulation comes from a discrete element solver of some sort, but we have all kinds of solvers for what resolution we want to solve the equations on, and you can produce a movie like this. And what I want to do is I want to figure out, can I find a spatiotemporal pattern of activity? I want to find an alpha of r comma t in such a way that I can drive the system to whatever pretty state I like. I'm a physicist, so my pretty states have to be kind of homogeneous, kind of nice, and stuff like that. So I can imagine that I want an ordered nematic domain to stay in my system forever, or I can imagine that I want to set up coherent flow so that whenever I introduce a tag particle into my system, it always goes from the left to the right. Those kinds of things is what I want to accomplish. Now, how do I do that? I use optimal control as a first step. And again, I'm going to give you the physicist's formulation of optimal control. Applied mathematicians should forgive me for the way that I'm going to formulate the problem. The way I think about it is, I have some PDE. I have some nonlinear PDE that is coupled, few variables coupled to each other in some way. And what I usually do as a physicist is that if somebody gives me the driving, the alpha, as a function of space and time, and tells me the initial condition, namely where did these fields start at time t equal to 0, what configuration they started in, I can solve for what these fields would be at any later time tf, right? I'm calling tf because it's going to be called the final time, in the sense that I want to ask what activity profiles do I need to have so that the system gets where I want it to go in a finite amount of time. Okay? This problem we can solve. This problem a physicist can solve in their sleep, by which I mean they can write the code and integrate it forward and figure out what's happening. But when I want to do the control problem, what I want to do is I want to solve for all alpha of r comma t in some time range 0 to t final, given the initial state of my fields and the final state of my fields. This is a boundary value problem in time. Physicists never solve boundary value problems in time. We are initial condition people. You can't ask where the Big Bang came from. We can tell you that there was a Big Bang. And from there, the rest of the universe evolved. Initial conditions are what we assume, and whatever comes out is what comes out. So we don't actually solve these kinds of problems, but of course, optimal controllers know how to solve this problem. And within control theory, you can ask, is it in principle possible? Can I in principle find an alpha of r comma t given the nature of my equations? So if I can find an alpha of r comma t for a given initial condition and a given final condition, then I call that would fall under the purview of controllability. And if I wanted to think about a manifold of initial states and ask what is the manifold of initial states, final states I can access from these initial state, that would be a problem of reachability. And these are all mathematically well-developed things. But for my particular fully nonlinear PDE system, I have no idea. And as far as I'm able to see, the literature also doesn't give me any idea about how to demonstrate either controllability or reachability. But I can linearize my system, and at the linear level, the system is controllable and a lot of states are reachable and stuff like that at the linear level. And so I assume, okay, it's probably locally controllable. I'm okay with that, right? So I stop there and I take a physicist for approach again. The proof of the pudding is in the eating of it. Namely, if I can find a solution, then the solution exists. I am not going to try to prove it, right? And the way that I'm going to do it is I'm going to write down a cost function. The cost function basically says, if you're not in the state I want you to be, then I'm going to be unhappy. So this guy is saying, this is the state I want to be in, and this is the state I am in. So if these two are different, the cost goes up. So the cost function basically says, I want to be in the end state that I want to be in. And I also put in an activity penalty because I want to implement my solution in the experiments. I know what range of activities the experiments can actually implement. So I want to limit my solutions to stay within an alpha twiddle, which is basically set by where the experiments can be. And I again add a penalty to gradients in activity because again, in my experimental system, there is a limit as to what gradients can be implemented in any realistic way. And so I have to penalize gradients if I want a solution that I can actually implement. Then I convert the cost function into an action by adding as constraints the dynamics that the fields should obey. So I introduce conjugate variables eta and nu. They are acting as Lagrange multipliers that enforce the dynamics of U and Q, right? Once I have an action, I'm a physicist. I can write down Euler-Lagrange equations in my sleep, and there are many, many techniques for me to solve my Euler-Lagrange equations. So what we do is we write down the Euler-Lagrange equations and use techniques that applied mathematicians have figured out. The first work that I'm talking about today uses a direct adjoint looping iterative scheme to solve the problem, but there are more fancy, much better ways that have been developed by applied mathematicians, and I'm reading, understanding, absorbing them into my scheme as I speak. So that's it. I implement this numerically. So that's the theory. What can you get from doing something like this? I just show you a couple of examples of solutions you can get from something like this. In the top part of the movie, you're seeing the velocity. The arrows are the velocity field. The colors are telling me the magnitude of the velocity. And the control problem I'm trying to solve is where there are these green dashed lines. I want deterministic flow of a given magnitude from the left to the right. Right? And in the bottom movie, the colors are associated with activity and these lines are associated with the nematic director. And what you can see is that my problem pretty much, these are for two different circumstances where we've changed some other parameters in the equation, but I'm able to find a solution for activity patterns that drive the flow that I wanted to drive, right? So I've actually managed to control the forces in some region in my system. And the only point that I want to note is that the optimal control solution for physicists is a little surprising because I'm not actually activating the system where I want the flow. I'm activating the system outside the region where I want the flow so as to be able to control the flow in there. There are some nice physics reasons why this is true, that activating outside gives you a better control of the flow in the region that you're trying to control. It's associated with nematics. I won't tell you that, but I think this is a very nice solution, which I did not think of before the optimal controller spit it out. So I was like happy that the optimal controller was slightly smarter than me, so I will use it. Okay. Because the first few things we tried, it gave me stupid solutions. All right, that I could think of. All right, and I just wanted to highlight these two papers that are basically implementing optimal control for two different kinds of active fluids. The top paper, I have no experimental system in which to test it in. The bottom paper has an experimental system, and I am trying to test it in there. I'll tell you in a minute what went wrong in my life when I tried to do that. But in the meantime, while I was building this whole infrastructure by saying that I'm going to figure out what the activity pattern is that I need to impose so that I can get whatever flow I want in my system, my experimental collaborators found a way to implement it in the experiments. What they did was they took the eyelid protein, which exists in many, many plants, and that makes the plant light responsive. It makes sure that your flower blooms when the sun comes out and makes sure that your plant bends towards the light source and things like that. They took the eyelid protein and stuck it onto our kinesin motors with some molecular tag whose name I'm blanking on in this moment. I think I'm getting nervous. I can't remember the name of the tag. Anyway, so it doesn't matter. It's a tag. It's a green ball. And what happens when you stick this green ball into the blue spheres is that the motor acts as a motor only in the presence of light. So the motor is basically completely inactive and not doing anything when there is no light. And when there is light, it starts acting as a motor. So now my challenge becomes slightly different. Oh, and they can use this kind of light pattern variation to implement closed loop controls in active pneumatic flows. This is not yet my optimal control solution. That's why I'm not on this paper. They did some PID control to test for for viability of this idea. Okay, and so now my challenge has changed. I think I have figured out the pattern of activity I need so the material will exert what force I want it to exert. But how do I figure out what this means for light patterns in the experiment? Because if I go to my experimentalist colleague, especially Zwanimir, if I go tell Zwanimir, here is alpha of R comma T, you have to implement in the experiment, he'll kick me out of his office and close the door. Because he absolutely wants me to tell him what motor concentration in micromolar per whatever I should put in, what is the digital light projector resolution you need, and what is the frequency that your digital light projector needs to be able to switch on, and things like that, right? So I really need to figure out what this means in terms of light patterns before I can get anybody to do it. So we took a machine learning approach to try to get to this. I will describe the baby version in this talk, and there is a fancier version that we're writing right now that will also show up soon. Okay, so what do I want to do? What do I know to be true at this point? I know that there's a length scale, and that length scale is a function of the ATP, the motor concentration, and the bundler, and things like that, the kind of numbers my experimentalists would want to know from me. And I could measure this number by using defect density and vortex sizes. And theory says it should be some proportionality constant that I don't know, and the square root of k over alpha. Similarly, time scale should also be a function of just these three molecular concentrations. And I should be able to figure out what the speed, it should be. And the proportionality constant times 1 over alpha is basically what it should be, according to the theory. So my proximate game right now is to say, I'm going to fix bulk ATP concentration, my KSA concentration, and PRC1 concentration, assume they are all homogeneous everywhere in my sample, and ask what is the intensity of light I need in order to produce a particular alpha in the experimental system. Okay, so that's my proximate goal. And the way we decided to do it was we say, okay, we'll take the movie from the experiment, we will compare it to the theory and try to figure out what these numbers should be, right? The experimentalists can produce data at different concentrations of ATP and things like that, and they can produce data at different light intensities and so on, and we'll just compare the movie to the theory and figure out what the coefficients are, okay? And the way you do that, again, this is work done with a former student, Chaitanya Joshi, and Mike Hagan, my buddy, is the lead on this project, and Pengyu Hong is our computer science collaborator who taught us some of the current generation machine learning tricks, which a very mathematical person like me cannot figure out. Because it's like, I have so much more math to learn, don't tell me about that yet. But he'll be like, no, you don't have to understand the math, just put it in there. Okay, all right. Let's start with the data. How do I do this? Compare the movie to my theory. I have some intensity, I of x, y, t. And I'm watching a fluid flow. So if an intensity of this magnitude is at this pixel, it's going to be here at the next pixel. I mean, at the next time step, because it's going to move. So the intensity has to obey an equation like this at some short enough time scale. So I can measure, I can fit to figure out what the flow velocity is. Similarly, if you think about it for a moment, this may be less apparent for people who have not thought about pneumatics, but the strongest direction of the intensity gradient will always be orthogonal to the direction in which the force dipoles lie. The force dipoles will lie along, the intensity will be more homogeneous along the lines than orthogonal to the lines. Okay, so I can find the maximum gradient direction of I locally and cross it with a layer normal, and that will give me the direction in which the director is pointing. So I can, in principle, extract my hydrodynamic fields from the data. Okay, initially, maybe 5-7 years ago, this is what I was doing actually to the experimental data. But of course, we don't do this anymore because we realize that what I'm doing is basically I have some problem like this. It's a matrix problem of some sort, and the matrix is sparse. We're doing physics. There's going to be lots of zeros, right? So this is a sparse regression problem. And if you're doing sparse regression, you should really not do it by hand anymore, right? So we built the machine learning solutions for this. We took the sparse regression approach. We showed the structure of the matrix we were expecting to our computer science colleagues, and they gave us a few neural network architectures that were most suited for that kind of data. And we built a machine learning solution that basically takes experimental data and converts it into the flow fields that my theory symbols correspond to, right? So instead of working with intensity, now I can work with my physical variables, the flow and the director. Okay. Then, to measure alpha from the experiments, we did the following. We said, okay, let's take the Q and V that I measure from my experimental data. Let us postulate the most generic PDE model you can write down for the system. So there's some time derivatives, there are lots of space derivatives, whatever nonlinearities you have the energy to write down. The limitation is only the patience of the postdoc working on it and the number of library terms you want to keep for computational efficiency. So you postulate some dynamics like this, you compute both sides from data, and again do sparse regression to try to figure out what those coefficients AI and BI and things like that are. Then I will have learned my model from the data. And in the paper that I will cite on the next slide, we used a hybrid of SIND and PDE find to build it. But since then, there have been better and better techniques that have come out that also we're adapting to try to do this better. Again, don't do sparse regression the stupid way, do it with machine learning is the lesson, and we're trying to implement that. And this particular technique, SIND and PDE find, I learned from reading these papers. So these references are not accurate except for the fact that these are the papers I read to understand what is happening here. And so what we did was we were able to find activity by doing this kind of a process. In the paper that I cite on the top of the slide, we did it it was not a light controlled system, it was a uniform activity system. But since then we have light controlled data and I can get to my goal of figuring out what this alpha guy is in my hydrodynamic theory as a function of intensity in units that Zwanimir would like, milliwatts per centimeter square, because that's how the digital light projector can control the light signal in their experiment. Okay, so now I know I have a procedure for finding out what alpha I need so the system goes to my target state, and I know what that alpha means in terms of intensity patterns in my experiment. So then I should just be able to take a solution and implement it in the experiment and it should all work. The world kills the theorist's dream. The proteins age. Material properties change as the experiment proceeds. It doesn't happen rapidly enough, but it happens enough that I can't control the system for any length of time with the solution that I started with. The second problem that shattered my dream was that the motors cluster. The initial motor concentration, which I assumed to be uniform, does not determine activity locally except at early times. The motors start clustering and you get clumps of motors. So you're looking at much more massive objects that need to diffuse around. And so my initial prediction doesn't hold for very long. But I still think what I'm doing is right because the statistical properties of the system change very slowly. I can still have robust vortex size distribution, defect density distribution, speed, etc. for order of hours. Those things don't change. So my physics is not wrong. It's just this numerical way of extracting alpha fails after a while because the assumptions that I put in my extracting of alpha fail, right? So what I realized is that I'm not wrong about what I imagine I can do. I just have to do it real time with the data, with the ongoing experiment. So I need to loop it around. I can't do like take 6 months, find the optimal control solution, then go downstairs and say, can you please shine this pattern and see what happens? That's never going to work, right? It works for the first 5 minutes and then falls apart, right? So I want to do it real time. So I pick myself up off the ground and make a new plan. And my new plan is the standard data-driven plan if you want to do anything in real time. The compression scheme is no longer the physicist compression scheme of minimal symbols. It's the minimal number of data points I can work with or things like that. And so we use a variational autoencoder to try to find the latent space representation for our data that is most robust. So far, we have succeeded in reducing the data to order of 10 latent variables. So I think our variational autoencoder is ready. It is suitable for our experimental system. Then I have to do forecasting. Again, I don't want to use the physicist compression. I want to use the machine learning compression. So therefore, I have to build neural ODs to do my forecasting. So my roadmap is the following. I have a microscope camera that sends me a signal that is literally completely uncompressed eye signal. That is basically the intensity that the camera sees in each of its pixel out to my variational autoencoder. And my variational autoencoder reduces the 10 to the 6 data points that my camera sends me to an order of 10 variables, right? Then I want to build a neural ODE that will predict from this compressed data set the next latent space variable that this system is going to end up in. And then I want to use deep reinforcement learning to figure out, basically I'm doing optimal control but not using the PDEs. That's basically what deep reinforcement learning is. So I do optimal control without the PDEs. I build a deep reinforcement learning network that will produce a solution for the control signal, namely the light intensity that needs to be shined on my guys to make them do what I want them to do. And so far what I have, and this will be the control signal to the experiment. So far what we have is the variational autoencoder, I think, is good. I like what it's doing. I kind of understand physically what the latent space variables are, though I'm not talking about them publicly yet because I need to verify what I have learned. And the other piece that has been built is the deep reinforcement learning. Since I know how to do optimal control, the deep reinforcement learning was the easiest thing to build. Neural ODE was harder to build because I didn't understand the math. So we built these two pieces. I'm in the process of building the neural ODE, and it's actually being built by a former Northwesterner, Aveg Agarwal, who used to work with Monica here. And he's the one that's teaching me how to build a neural ODE, and we're still fighting. I don't believe him, so we'll figure it out soon. Good deal. And okay. And I will remind myself, never forget the goal. I want a bag of crap that goes where I want it to go. So I want to remember that that's where I'm going. And to that end, I've been talking to my experimentalist colleagues about bags and if we can understand the bags. So I want a bag inside which I want to put my active entities. So what kind of bags can we make? We can make an oil water emulsion bag. So this is a bag in which there is a water droplet that's enclosed by a layer of oil. And that oil is the material bag that is holding my active material inside. So we've done some preliminary studies with this bag. The first paper came out last year, but this is still very preliminary. We don't understand this bag yet, and this bag is also kind of boring. So, and this work was done by Salman Alam, a graduate student in Guillaume Duclos' group. And there's another bag, which my experimentalist colleagues have built. This is an entropic bag. What this bag does, this is work that is being done with Guillaume Duclos and Zvonimir Dojic. Zvonimir's first couple of papers are out already, and Guillaume's papers are getting written right now. And what this bag is it's a phase separation, liquid-liquid phase separation bag. So what I have is I have two kinds of polymers in my system. I have PEG and I have dextran. They are both polymers suspended in the fluid. And I don't have to worry about viscoelasticity and nonsense like that. There's very low concentrations of these guys. But the microtubules like dextran. So they will automatically go and sit on one side of a PEG-dextran interface. That's basically what you're watching in the movie. This is a PEG-dextran interface, which is an entropic interface. It's a phase separation interface between two liquid phases. But that entropic interface encloses the microtubules, and you can watch the fluctuations of the interface. This is the bag that I like more. and we're playing with this bag to try to understand if I can, make my thing do what I want it to do. Okay, and there's a third bag. This I don't have at Brandeis. This is a bag that Andreas Bausch's group has, and Petya here at Northwestern knows a lot about. This is a unilaminar vesicle bag. So they use a sea dice system to make unilaminar vesicles inside which they put our microtubules and motor proteins, and the bag sloshes around in various interesting ways. And this is also not yet published, but I heard that the paper is out the door, so you should see it soon. And okay, and I want better things to put in the bag. So my experimentalist colleagues have been building better things to put inside the bag. The microtubule system is a little finicky, so what they did was they embedded the microtubule system inside a passive polymer network made out of actin. Then you can get this Godzilla type dynamics happening in the system. The actin forms an elastic network of some sort, which is getting distorted by the microtubules. So I really like this elastic stress propagation motif that is here, and we are working to try to understand what is a good theoretical way to work with this. I haven't looked at the data on this problem yet because I want a theoretical picture that makes sense, and I don't have that yet. Another system, and this we have made more progress on, where the experimentalists have put the microtubules inside a liquid crystal. The liquid crystal is made out of FD viruses. FD viruses left onto themselves form very nice liquid crystals. At high densities, if you put enough of those viruses, they form very nice liquid crystals. And what the experimentalists do is introduce the microtubules inside the liquid crystals. This actual Z-stack movie from the experiment obtained with laser sheet gives me three-dimensional information about what is happening inside my system. This raw movie, I don't know how to work with yet, but with a little bit of analysis on the movie, you can reduce this movie to topological structures that are highlighted in this picture here. We can see disclination lines, and we can see topological loops and things like that. And these are the objects that I will seek to pick out from the data and use that because once I see this object, I know what the stress is there. And I can use that to try to figure out how to control these systems. These are all pie in the sky goals. 5 years from now is when I will know whether any of this is going to work or not. But the theory seems like, oh, it's really good. There's a nice hydrodynamic theory. Everything works and so on. Okay, good deal. Finally, can I go for another 2 minutes? Yeah. Okay. I just want to express, I'm an eternal optimist, right? I keep falling down and bumping my head and against hard objects and stuff like that, but I still think it's all going to work and I'm going to go where I go. So I want to give you my rosy future. So I want to build this bag, right? And there are all kinds of problems with the materials that I'm using to try to build this bag. So I'm making friends with people from different fields, talking to synthetic biologists because they have a way of implementing bespoke biochemical cycles inside a cell, right? I want to take some of those tools so that I'm not controlling things with light from top down, but I'm controlling it with reaction diffusion inside the system, right? I'm optimistic about that. I'm talking to de novo protein design people who know how to work with AlphaFold 3 and things like that, and make them make proteins that I can use, right? Rather than go hunting in nature. What we've been doing so far is go hunting in nature to try to find the protein that would do the right thing for us. Maybe we don't have to hunt in nature. Maybe we can modify the protein we already have so that it is not sticky, that kind of thing. So I'm optimistic. And then there are all my friends in active matter who are trying to sort out the mechanics and energetics. They are basically trying to figure out the organization principles in space and time for these driven systems, internally driven systems. And I hope to learn from all of these people. And of course, broader out, I don't have that many friends in these fields, but the biochemistry, cell biology, and organismal biology is filled with people doing very, very clever things. I want to borrow all of this and use all of it to build my bag of crap. Okay. And of course, machine learning and data science sitting in the middle makes most of this possible, because otherwise this is way too complicated for my simple mind. All right, finally, acknowledgements. As I mentioned, I don't actually do anything. I go to random people's offices and have conversations. And these are the people who do the work. Zvonimir Dojic at UCSB, Seth Freden at Brandeis, and Guillaume Duclos at Brandeis are the experimentalists. Mike Hagen is my computational guru. And Pengyu keeps us from burning ourselves too bad by picking the wrong neural network. Right. That's everything I wanted to say. I will stop here and answer any questions that you might have. Thank you.
We have time for a few questions. Any questions from the audience? I can start things off. if you guys are thinking about them. One question. So I have one very quick concrete question. What are the microtubules in the experiment? Are those biological or are those
They are biological, purified proteins. So we purify tubulin, and tubulin polymerizes into microtubules, and there are ways to truncate the polymerization at selected length scales. So we have some measure of length control over the rods, but they are purified, and it's purified from the brain of a kosher cow to get the thing. And apparently tubulin prep only works at Brandeis. Anybody who has this material, we ship the tubulin and the motor proteins to people.
Interesting. So then, sort of on a totally different topic, but then I asked that because I wonder if you can if that could be replaced by something totally synthetic, but maybe that's not a priority.
So we are trying, we haven't tried totally synthetic, but we are trying to make it work with DNA nanorods. So DNA origami nanorods. So take DNA and fold it into a nanorod. You can do that reasonably easily. Then functionalize the DNA with certain peptides that can stick out of it and talk to the rest of the stuff. It hasn't worked yet, but in principle, I don't see why not.
Here's maybe a more conceptual question, and I'm sure you've thought about this. So your feedback control right now, or your plan, is to have the full kind of image information as the input, but towards your broad goal of designing your own
bag,
amoeba or whatever that's going to control itself, it will only have local sensing information.
Yes.
So have you thought about how you might deal with that problem?
Yes. So the way, I don't know how to do it for my moving bag of crap yet. I'll have to make it move with light to begin with, but I can control some other things locally. So one of the things that you can do is you can take the active hydrodynamics like I describe, or one of those theories of that oeuvre, and couple that alpha to a reaction diffusion system. So that alpha is the output of a reaction diffusion system, right? And then that reaction diffusion system can have characteristic length scales, time scales, patterns associated with it, which will drive the activity up there, right? So that kind of thing that you can do, that will be really local control. I can't yet do it at the level at which it can't be the first step, because I'm still trying to figure out how the reaction diffusion couples to my hydrodynamics and what are the things I can or cannot do. And I don't have a good picture yet, and the first experimental system is just beginning to take shape. So I'm hoping that that would be the thing. And eventually, if I wanted to make that bag of crap move, if I go and look in the literature associated with that bag of crap that actually exists in our blood, the reaction cascade is like 1,000 reactions big, right? There's a reaction cascade of, ARP23 getting recruited to the membrane, and then something else coming and making ARP23 happy, and then ARP23 recruits F-actin and whatever, so on. So the reaction cascade is like 1,000 reactions strong. I think that by starting with this macroscopic goal, we could get there in the order of 5 reactions, let's say, or 6 reactions. I don't know. We'll see. Right now, we're just trying 1 reaction, and it's already complicated in the experiment. In the theory, it's very pretty, but in the experiment, it turns out to be, Yes, sir.
Yes. So like following by the back thing, but so in your simulation, you show the beautiful simulation, the boundary condition is repeatable, right? It's
periodic, yes.
But in your, can you elaborate, how do you define the boundary when you have different emotions or, then.
Numerically in my computer.
Yes.
So we have a few different schemes. So you can do phase field. That's the easy, cheap and best. And you can also do immerse boundary. So those are the 2 schemes I have. I have an immerse boundary method that can track grid points where I decide the interfaces, and I can assign the grid points whatever mechanical properties I want, the stiffness and the bending modulus and things like that. And in the phase field, the interfacial tension lambda will control both of these. And so you can use a phase field model as well.
Thank you so much. Thank you for the great talk. So I have a question regarding the compression part with variation autoencoder. So for that complex system, I'm wondering, when you get the optimal control from that method, what are the variables you're getting from that method? Because in that case, you're You don't have like physical
Yeah.
parameters.
So I always start by training my model on synthetic data where I know what flow is and what Q is and things like that, and make sure that what it's spitting out is the alpha that I would have gotten if I had done it the old-fashioned. Then when you're doing it on experimental data, you train it not on the intensity data. I lied. You put a machine learning layer in between to make it into flow and Q data. So there's a machine learning layer in between that takes the intensity data and converts it into physical data.
I see.
So there's an optical flow code that converts it into velocities, and there's a little some LSTM forecasting thing that converts it into Q, and those two I hid. I did not show you. That's why I know what comes out is the alpha that is in my theoretical model, because I'm giving it my theoretical variables.
I see. Like you have like physical structures in the machine learning model.
And I'm working on learning what the latent space is teaching me. I cannot do it for the full 2D system I talked about today, but I can kind of do it when I have a 1D system. So I can watch the interface, right? I can take a very long flat interface and watch interface fluctuations and do variational autoencoder on the 1D thing. I'm a physicist. The interface can only have a few simple terms in its dynamics, right? So whatever that autoencoder is learning has to be some Fourier coefficient or has to be a moment. So then you start clustering your autoencoder data to try to see which ones those are and things like that. And it seems like it might work, but you can't hold me to that because I have not written the paper yet. It may all be wrong tomorrow.
Yeah. What's the original dynamics in 3-dimensional space, or it's originally just in 2-dimensional space?
In the problems that I described in today's talk, they are all in 2D. For the interface problem, I'm just tracking the fluctuation of the bumps and valleys, right? It's a 1D height field. It's only a function of x and t. And you also have this real 3D data that's obtained from laser sheet and confocal and stuff like that. That's really 3D, 3 space directions and 1 time direction. So it's like enormous.
I see. Thank you so much.
Okay, if we don't have other questions, then let's thank Aparna again, and thank you all for coming.